Hey everybody, and welcome to our very first lecture in our online microbiology series. Our lecture one is going to cover the introduction to microbiology, kind of what microbiology is, kind of a couple of the big fields in microbiology, a couple of the big famous scientists in microbiology, and a couple of things like that. So let's go ahead and get started. Well, first off, since this is a microbiology course, we need to talk about what microbiology is. So microbiology is the study of things that you can't see with your eyeballs, tiny little living things, hence the name microbiology. There are about 6 or so things that are generally classified as the microscopic things that we study with a microscope. There's going to be a couple of eukaryotes, one prokaryote, and one couple or maybe 2 or 3 other things that are acellular, but usually just one that's focused on regularly. Fungi, helminths, algae, protozoa, that's going to be our eukaryotes. Our prokaryotes are going to be bacteria, and our acellular is going to be viruses. And we'll talk about those a lot in this course. There's a couple pictures of them over there in the corner. So why do we care about microbiology? Well, microbiology has killed a lot of people throughout time. Most of the deaths during war, up until very, very recent, have occurred from infections that have occurred either off the battlefields, people living in tents and cramped conditions and things like that, or infections that they get from some wound that they got on the battlefield and things like that. Millions of people have died from things like bubonic plague, smallpox, common cold and flu viruses and things like that have killed millions of people. Surfaces of the earth, continents, and in the sense of who owns them, and governments and things like that, civilizations have really changed solely because of microbes in the microbiological world around us. In fact, we've even used it in warfare dating back for a really long time. You can see that picture up there of soldiers launching dead bodies that are plague victims over the walls of castles that they're besieging, so everybody inside gets sick and then it's easier to take over. Today there are quite a few fields in the microbiological world that you can look at if you're interested in studying microbiology. You got the public health and epidemiological field. This is going to be people that work with the governments, local, federal, state, and things like that. Kind of coordinating disease response, coordinating vaccine delivery, getting antibiotics out, figuring where diseases might be coming from and how to control them, implementing quarantines and things like that if a particular disease is detected in an area. Federal, state, and local governments and things like that, coordinating the activities between them. So pretty cool on that one. We got biotechnology, and this is people that use living organisms in the sense of cells and single-celled organisms, yeast and bacteria and things to do something for humanity. And this can be anything from like making cheese, brewing beer, and stuff like that, all the way up to big stem cell research where you're using them to regenerate organs for people. You've got environmental microbiology, and this is looking at the relationship between the organisms that live in the soil. There's quite a few billion of them that live in the soil. And they're all impacted by what we do as well. If we have sewer runoff or agricultural runoff from water, or if we put pesticides or fertilizer on a piece of land for food, that causes the microbes that live there to have different reactions, and that really needs to be taken into account, especially because sometimes sewer runoff can cause bad reactions for people's drinking water and things like that, or contaminate crops. You've got immunology, which is the study of how the body deals with invaders like microbes and fungi and things like that inside of us. And this is going to be trying to develop vaccines or developing allergy tests, all kinds of cool stuff. You got genetic engineering, and this is the one that everybody likes, where you take an organism and you alter its DNA in some sort of way to make it do something new for humanity. Either producing some new product or something that they didn't do originally. Just really cool little products. You got agricultural microbiology, and this is looking at the relationship between the things that we eat, plants and food, and animals and stuff like that, and the bacteria and microbes that live on or in them. Just like us, they have bacteria that impact them, that make them sick, plants and animals, and the antibiotics that we give them to treat those have impacts on those populations of bacteria and microbes and things like that, so you have to monitor that kind of stuff as well. And then the last one, and last big field, this is probably the most common, that's always looking for jobs, is people that are going to be working with food microbiology. And this is people that work in plants that are like going to produce commercial food plants and things like that. or you go into like a Mars pet food or the Hershey plant, and you swab all the surfaces that food comes in contact with, and you make sure that everything's clean and properly sanitized and that they're following the correct methods. So if something does enter their plant, nothing will or should, but if it does, it's controlled and maintained, or if it does enter the plant, it's reported properly and things like that, and to try to minimize the risk of contamination. So that's a very, very important job that's always looking for microbiologists. If you're interested in being a microbiologist, that's a good one to go into. Well, as I mentioned, I'll elaborate just a little bit more on these guys. There's really no line of what you should and shouldn't study if you're a microbiologist, but it's just stuff that you can't see with your naked eye. But there are quite a few things that are kind of big that are tossed into the microbiological world, and some of them, their larval stages or their eggs might be microscopic, and that's why we talk about them in the microbiology studies. And then their adult form might be quite big and macroscopic. You can see it very easily. But for the most part, these are the couple of eukaryotic cells that are tossed in here. We got yeast down here on our bottom left down here. We've got an algae, a paramecium, and a worm, a helminth up here in the top. And some of these guys are quite large. You can see them with your naked eye. Some of them you really need a microscope to look at. But for the most part, if you want to get good detail and see these things in reality and see what they're really up to and what's going on inside of them, you need a microscope. And that's why they're kind of tossed into the microbiological world. We also have a couple of members of the microbiological world that are not living. Viruses you're probably very familiar with. Things have to have a cell to be considered living, and these things don't have cells. We'll talk about that in a little bit later. But viruses, viroids, prions, and things like that. Viruses you're very familiar with. It's a little piece of genetic material, DNA or RNA, never both. That's a key characteristic of viruses. They have one or the other, never both. DNA or RNA, never both. Surrounded by a little piece of protein. Viroids are plant problems. They are essentially just a little plant virus that doesn't have a protein coat. And then a prion, is a misfolded protein. They have the ability to cause problems in plants and animals, but they're not living, and that's super weird, and we'll talk about these a little later. Generally, this is kind of what we study in the microbiology world, tiny little stuff, up to about where the naked eye level starts. Animal cells, plant cells, you really need a microscope to see them quite well. You can see frog eggs, you can see chicken eggs, you can see people, things like that. You really need a microscope to get down into the inner workings of a bacteria. You can't see those with your naked eye, and things like that. You really need a microscope to get down into that small. But this is kind of the broad spectrum of things that we kind of look at in the microbiological world. In the spectrum and what you really need a microscope to see. And we're going to focus on stuff that you really need a light microscope to look at. Viruses are a little farther out of the light microscope range. You really need an electron microscope to see them really well. But we'll talk about them simply because they're just so important to microbiology. Well, I've mentioned the term prokaryotic and eukaryotic a lot. And what does that mean? Well, cells are living things. You've heard the term cell before. You probably know what a cell is, or at least the idea of a cell. There are two different types of cells. There's prokaryotic cells and eukaryotic cells. Now, prokaryotic cells, what does that mean? Well, pro means pre, karyotic means nucleus, so things without a nucleus, before a nucleus evolved, essentially. Whereas eukaryotic cells, eu meaning true, karyotic meaning nucleus again, so true nucleuses, things with a true nucleus that evolved a little later. All bacteria, and as well as archaic cells and some types of algae cells called cyanobacteria, are all going to be prokaryotic. They have unique traits that are found within them that distinctly break them apart from our eukaryotic cells. Whereas our eukaryotes are going to be the early ones I talked about, helminths, fungi, algae, and things like that. And they evolved a little later in the fossil record. And if you just had a microscope and somebody gave you a slide, the easiest way to tell apart the two different cell types, if you just looked at it and saw a bunch of cells in there, and one of them was really big like this and one of them was really teeny tiny, prokaryotic and eukaryotic. The size difference between the two is just insane. Prokaryotes are teeny tiny. Eukaryotes are huge. So if this was a prokaryote, I'd be a eukaryote. That's the size difference. They're just drastically different. One of the main differences between just the size, more the important details, size is just kind of trivial, it's just there. They're more complicated on the inside, so they need a lot more space to be more complicated in. But essentially, the true nucleus part, the eukaryote, the nucleus there is that big deal. It's a protective control center for the DNA. It kind of keeps all the DNA shielded and safe from enzymes that are floating around out inside of the cell that might damage it or destroy it, and things like that. Whereas prokaryotes, they don't have that. Their DNA just kind of floats around inside of their cytoplasm, kind of in a big blob sometimes. And that's the difference between the two. That protected DNA or that kind of naked DNA that just floats around inside of the cell. Now, prokaryotes, on a more detailed level, all prokaryotes are unicellular. Unicellular refers to organisms that don't need other cells to exist around them. They don't need other cells to do anything at all. I'm a multicellular organism. Most of the things you're probably familiar with are. And we need other cells around us to function. If I pulled my liver out, it'd be a blob of liver cells, put it on the table. I would die because my liver wouldn't function, and my liver would die because it wouldn't have me to keep it functioning either. Whereas a bacteria, it does everything it needs inside of its one cell. I can't function as one cell. My body can't function without the rest of my cells. We depend on one another to function, and these guys don't. They can reproduce by themselves. They can do everything they need by themselves. They don't need a partner. They might live together, but they don't need a partner to do anything. Sometimes it's easier for them to live together, in the same concept of, it might be cheaper to split the rent and things like that. They can find, it's a little easier to get protection in numbers and concepts like that, but they don't need a colony to live in. They can do everything they need by themselves, and they're all microscopic. They're all teeny, teeny, tiny. No membrane-bound nucleus. Like I mentioned, they might have a blob of DNA that floats around in the middle of their cytoplasm. DNA sticks to one another just based on its chemical charge. It blobs up together. Looks like a little yarn ball called a nucleoid. And sometimes they have that, some of them don't. They have no membrane-bound organelles inside of them. They basically are just a mitochondria, so they don't have mitochondria. They don't have chloroplasts. They don't have Golgi apparatus. They don't have endoplasmic reticulum. None of that stuff. They basically just are a giant mitochondria. We'll talk a little bit about that later. One of the unique things about these guys is they have cell walls that are made out of a unique sugar protein complex called peptidoglycan, hence the name peptidoprotein, glycan, sugar, that makes up their cell walls. That's not found in eukaryotes ever, ever, ever. No eukaryotic cell on the planet produces that, so it's unique to prokaryotes. That's a very important thing, and we'll talk about that later. These guys almost always reproduce by a binary fission, which is just asexual reproduction in the sense of, boop, made a clone of myself. One turned into two, and they're identical copies. We'll talk a little bit about that later. Useful in some circumstances, not so useful in others, but that's kind of what they do. They can sexually reproduce, sort of, kind of, but they usually don't unless they just have to. And they have a very unique type of ribosome that's found in a prokaryote called a 70S. Now, we have ribosomes. Ours are 80S. Theirs are 70S. It refers to S means Svedbards, how fast the centrifuge spins around. It's kind of neither here nor there. It's just kind of one's bigger than the other, basically what that means. So they do the same thing. They make proteins, but they're, one's bigger than the other. So prokaryotes, there are two different main groups of prokaryotes, termed domains. You got eubacteria, true bacteria, you again, and archaea. So eubacteria is things that you're probably familiar with, are things that make us sick, going to be E. coli, salmonella, staph, stuff like that. And then you've got archaea. And archaea are weird little guys. And these things are like methanogens, halophiles. They live in these weird places that are either really super salty or really hot, really have low pH, really high pH, just awful, awful, awful environments where most things can't survive. And these guys are going to be really happy to live there. They're very similar in the sense of they have peptidoglycan, stuff like that. But the differences between them are very important, and it's what allows them to live in those different places. If you and I tried to go down into a hot spring, our cells would melt. Most bacteria would too. So they need to evolve a little differently so their cells don't melt, and that's the differences between them. Now, eukaryotes, much, much, much bigger, like I mentioned. And eukaryotes can come either unicellular or multicellular. Multicellular organisms depend on the rest of the cells around them to keep themselves functioning. One cell cannot live by itself and do everything that it needs. So it needs the rest of the cells, the organism as a whole, to function. Now we have these true membrane-bound organelles inside of eukaryotic cells. It's basically little rooms, if you like, that are specialized to do things. Mitochondria makes energy. The Golgi apparatus sends stuff around the cell, stuff like that. Right? Little specialized categories. We're a whole lot bigger. These types of cells are a whole lot bigger, so we need to categorize stuff off inside of them. The way they divide, not always by binary fission. There are a couple of them that can, but for the vast majority of these guys, they're going to divide by mitosis and meiosis, which is a different process. You've seen that in biology before, where the chromosomes line up in the cell and they split apart and things like that. True membrane-bound nucleuses, bacteria, prokaryotes, they have, for the most part, circular chromosomes that are round. Our cells, they link on the other end. Our chromosomes are going to be some lines, and you've seen that before, the little X thing before. The cell walls in eukaryotes are going to be made out of chitin or cellulose, depending on if you're a plant or a fungi. No peptidoglycan in us. Much bigger again. And then a slightly bigger ribosome called an ADS ribosome. So the little Venn diagram down in the bottom down there shows the commons between the cell types and the differences between them as well. So protists, kind of a weird little critter. They're all technically eukaryotes, but they have a bunch of primitive, very similar to prokaryotic functions. No peptidoglycan in these guys, but they may have mitochondria that doesn't really work, or chloroplasts that don't really function, or sort of like half-evolved mitochondria. They're just weird little critters. Just strange. So don't worry about these guys too much. But a lot of these guys do cause diseases in plants and animals, and when we get there, we'll talk about them a little more importantly when we get there. So just kind of be aware that they exist, just kind of weird little critters. So let's talk about the discovery of microbes, kind of where the people started to kind of figure out that these things existed. So it makes sense if you think about it that people really don't or aren't still aware of things that they can't see, and it's hard to imagine thinking that there's something out there that's microscopic that you can't see that can somehow do something. But for a long time, that was kind of just accepted reality. There were things out there that we couldn't see, and nobody cared, nobody knew they were there, and nobody thought to look. Well, eventually people started to get curious and people started to look, but they needed tools to look with, and microscopes weren't really that useful yet. Until about the 13 or 1400s, people really weren't aware of what was down there, these little teeny tiny things. And this guy came along, Robert Hooke. He was the very first person to look at something underneath a microscope that was a cell. And he didn't know what a cell was. We didn't know what cells were as humans. He was the very first guy to look at one underneath a microscope and kind of figure out what he was looking at. And he kind of came up with the term as well, a cell. He took it from a monastery, prison cell, same kind of concept, a little place where people live that has everything they need inside of it. And he looked at this piece of cork down here, took it off of a piece of wine bottle, cork, and slivered it and put it underneath his microscope, and saw all these little square cell walls inside of it and went, these little things, these little tiny rooms must be a plant, little tiny individual plants inside each one of these little rooms, and he called them cells, and that's where the term comes from. So Robert Hooke was the very first guy to do that. The next guy, kind of the big breakthrough in microbiological sciences, was Antony von Leeuwenhoek. So people had now kind of figured out that there were these tiny things sort of out there, but they really weren't, that aware of them. And this guy was the very first person to look at living stuff underneath a microscope. Now, he made his own microscopes. He was a Dutch guy. He was not a scientist, just had a lot of money and a lot of free time. And he liked to tinker and make things. So he made this microscope out of his own design. And he took a piece of lake water, a little drop, and put it on his microscope, and he saw all these little things moving around inside of it. Now, he termed them animalcules. You can see a drawing of them down there from his original sketches. And he took these notes of what he saw. He took these really detailed drawings of them moving around and eating and swimming and stuff underneath his microscope. And he was the very first person to ever do this, to see these living things underneath a microscope. His microscope was just so much better than everybody else's at the time. And he could see stuff that no one else could. So he sends a letter telling the scientific community, hey, look what I found, and nobody believes him because their microscopes weren't good. They just couldn't see anything. Nobody believed him. Eventually, he keeps pushing. They finally accept that, hey, there's tiny little things out there, and the world takes a little step forward where we finally realize that little things exist. Well, now that we've realized that these little things exist, we need to explain not only where little things come from, but big things as well. And you've probably heard the concept of spontaneous generation, where life just kind of poofs into existence kind of out of nothing. Well, this guy right here, Francisco Redi, really wanted to disprove that idea. He thought it was kind of silly, just kind of makes sense if you observe anything around you, like the reproduction of farm animals or humans or anything. And he really wanted to show that life had to come from other living animals. So, you brought other humans into existence. Flies brought maggots, which brought forth flies into existence. And that was kind of one of the big main things that people thought. Rotting foods, rotting meats just kind of poofed flies into existence. Maggots just kind of came out of the rotting meat for some magical way. So what he did was he took these jars here. He put gauze over one, left one open to the air, and left one completely sealed where no air could get in. The steak started to rot. Maggots showed up in the open one. Steak started to rot. The maggots showed up in the one covered with gauze, but they couldn't get inside. So they laid their eggs on the outside, and the maggots showed up on top of the gauze. Kind of showed where the maggots came from. And the one that was sealed with with parchment paper, no maggots showed up. The steak rotted. No smell at all was out into the air, so the flies didn't show up. So he showed right there that flies led to maggots, and you had to have both to get one or the other kind of thing. But what this did was it just showed that big things didn't poof into existence, but little things could poof into existence. Because what happened was the air, as we now know, was contaminated with tons of mold spores and bacteria, all kinds of fun stuff. And as he closed that parchment paper over the jar, the bacteria that was stuck inside of the air got on and fungus and all that stuff got on the steak, and it still rotted and went moldy. So people went, flies don't come from nothing, but these tiny little microscopic things, they do. So we moved from spontaneous generation to spontaneous microbial generation. One step kind of forward and a little step back. And then people started to figure out maybe it's in the air. So they tried their best to sterilize the air, sterilize the flask, combinations of the two, until eventually this guy right here came forth to figure out that, hey, it's in the air. I'm going to show you it's in the air. I'm going to show you how to keep it out, and I'm going to prove that if I put it from the air back into the stuff that's clean, and I can link it together, that's really what's going on. So Louis Pasteur, really, really, really smart guy. So what he did was he essentially took a toilet paper tube and he shoved it full of a bunch of cotton. And he sucked it through the air and he filtered out the air. And he took some notes and took some dust that he collected out of the air and he stuck it underneath his microscope and he took some notes of what he saw. He drew it and things like that. And then what he did was he had these special little bottles invented, and a French glassmaker made them for him, where it has this little hollow tube that comes out. You can see that there, called a swan neck bottle. It's essentially just a straw, where air can get in. Bacteria and stuff that's floating in the air can theoretically get in, but the bottom is lower than the top, so air pressure can't push up very high. It's a roller coaster concept. So bacteria and things that are heavier than air will sink and get caught at the little crook in the swan neck bottle, and that's exactly what happens. So what he did was he put some food for bacteria in it, stuff that they would love. Broth is what we know it as. Broth comes in two different flavors. You have broth made out of animal broth. We understand that today, chicken broth. And then you have vegetable broth. And that's what they had back in the day. He just happened to use animal broth. Very important detail here. So he sterilized his animal broth. He boiled it, killed all the bacteria and stuff inside of it, and he left it sitting out into the air. Sat there for weeks and weeks and weeks and months and months and months and years and years and years, and there's still one of these sitting in the Louvre Museum today in Paris, or the Louis Pasteur Institute, that's still sterile, which is kind of cool. Well, anyway, eventually what happened is the little crook, the lowest part where the bacteria started to, couldn't go up any higher because of the air pressure, they started to collect there. A little dust layer started to collect. So he knocked off part of that, took a scoop out of that dust, stuck it underneath his microscope, and looked at it. And lo and behold, it's the exact same stuff that he filtered out of the air. So, hey, the air, stuff collected there. There you go. So what he did next was pretty smart. He took another bottle, he tipped it forward, and he collected a bunch of that dust into that thing, from the swab, the neck, into his broth bottle. And his broth bottle that had been sitting there for months on end, open to the air, sterile, the second that he touched that dust in it, it instantaneously got contaminated. And a couple of hours later, it was all nasty and gross looking again. So he was able to show that there was a link between the air, stuff in the air, and the contamination of something. And as long as you kept stuff out, and he could show where that stuff came from, that it didn't just poof into existence in that broth. It had to be brought in from somewhere else. But lo and behold, the fact that he used animal broth was kind of also bad for him. Vegetable broth, unfortunately, was also very commonly used at the time for microbiology broth, and you can't sterilize vegetable broth very well. We can nowadays, they couldn't. It turns out there's something in vegetable broth called an endospore. We'll talk about those a little later. It's a heat-resistant form of a bacteria. It's not present in beef broth, animal-based broth. So if you use vegetable broth, they couldn't sterilize it. We can now. Louis Pasteur used animal broth. His was nice and sterile. So people tried to replicate his experiment, and they couldn't if they used animal broth, or vegetable broth, sorry. You just couldn't get it sterile. Could not get it to be sterilized. It would look sterile. It'd sit for a couple of hours. The dust wouldn't even collect yet, and it would just instantaneously go bad because the bacteria weren't killed when you cooked it, sterilized it to start with, or tried to, and they just started to grow again. Well, anyway, they didn't know that yet. They didn't know that these endospores existed. Eventually, they figured it out. This guy right here came forth, figured out, put forth the idea that maybe something was out there that was explaining that, these endospores. And then eventually this guy, Ferdinand Cohen, finally found these endospores in 1876 and put to death that stupid theory of spontaneous microbial generation. Took quite a while to get there, but finally that we got there. People figured out where these things came from, where they were going, how they got there, and essentially how to kill them at this point in time, and connected where they came from, how to keep them out, and stuff like that. So really now we can start looking for how to cure them, how to control them, how to prevent them, how to stop them, and stuff like that. And that's really what happened for the next couple of years or so, couple of decades, called the golden age of microbiology. This is when people started to really have functional microscopes. They really started to figure out what bacteria caused what disease, how to treat some of these things. No real antibiotics yet, but they were kind of starting to figure out the idea of them. They kind of started to discover viruses at this time period. So they were really getting some big breakthroughs in the late 1800s, early 1900s in the fields of microbiology. So smallpox was eradicated in 1977 through a worldwide vaccination campaign. Polio, mumps, rubella, measles, diseases that our grandparents had, great-grandparents had, the bubonic plague that killed millions of people in the past pretty much are non-existent anymore. They are still around, but nowhere near as devastating as they used to be. So microbiology has done a really good job at eradicating diseases that wreaked havoc on our communities at one point in time. So the future of microbiology is really cool as well. There are ever-evolving diseases that still need curing, Ebola, Marburg, and things like that. If they ever evolve the immunity to go airborne, they're going to need some serious vaccine research and treatment to develop real fast. Genetic research, developing new treatments for things, environmental cleanup. We can make bacteria now that eat oil. So if we have another oil spill in the oceans, we can dump gallons of bacteria in there. They can just go gobble up all the oil and turn it into something useful for the environment. So really, really, really cool things that can be done in the future of microbiology and things that are going on now. So if you are interested in being a microbiologist, check some of those out. Some of them are really interesting. So now that people have kind of figured out what a microbe is, where they come from, things like that, people started to put together what cells are. Once we kind of started to figure out all the idea of a cell, they needed to really define what a cell was. You need to have a line drawn somewhere that says this is and this isn't a cell. Because if you don't, it's going to be difficult to figure out what to study and what not to study and what to lump into the field and what not to. So these guys drew the line. They came up with something called the cell theory, Schleiden and Swan, two German scientists. And this is the cell theory. Now, the first three are the original cell theory. And this is what they defined as a cell. If you have these traits, you are a cell. If you have not these traits, you are not a cell. If you have any of these traits, but not all of them, you could have 4 or 5, but not all of them, you are not a cell. You have to have all of them. And this is the key characteristic of cells. This is what makes a cell a cell. Now, the first 3, all living things are made up of at least one cell. If you're a cell, you're a living thing, you're made up of at least one cell. You can be one cell or more cells. That's just the key characteristic of a cell. Every single living cell on the planet arises from another pre-existing cell by some form of cell division. Now, either nature broke its first rule and brought forth into the existence the first cell, or some supernatural being. Doesn't really matter. We are here. The cell is the fundamental unit of life and structure. Now, that makes sense if you think about it in the sense of I am made up of collective cells. Collectively, I'm a bunch of cells. So it's making me up. So fundamentally, I am cells. The cells moving make my arm move, make my muscles move, which make me move. So without them moving, I wouldn't be moving. So collectively, I'm just the sum activity of all of my cells, kind of thing. And that's the next one. The sum activity of all the cells depends on the sum activity of you. If 50% of your cells don't work, you're not going to work either. Or if half of your one cell works, you're not going to be able to do but half of your work that you normally could. So the first 3 right there are the only ones that they were aware of, the only things that they could come up with. The rest of the stuff, they weren't aware of DNA and RNA and metabolism and stuff like that. So the other 4 were added later. So energy flow, metabolism, photosynthesis, the production of ATP only occurs within cells. The only thing that can use and make ATP. Cells contain both DNA and RNA. That's a big key characteristic that I mentioned earlier. Viruses have one or the other, never both. Cells always have both. That's a key characteristic of living versus not living as well. And basically all cells are the same between very closely related species. And that makes sense if you think about it. My cell is probably very similar to a chimp, but not that similar to a bacteria because I'm not that closely related to it. Which also makes perfect sense if you think about it. But Staphylococcus aureus is probably very similar to Staphylococcus epidermidis. Their cells are probably exceptionally similar, even though they're very different from mine. So that makes sense if you think about it. So this guy right here, Robert Koch, another big scientist, big name in the microbiological world, was the very first guy to figure out how diseases are connected in the sense of this particular organism causes this particular disease. We still use his methods today. They're called postulates. It's really cool. Essentially what he did was he went out into a field of sick animals or something like that and identified a pathogen or an organism that was found only in the sick cows that the healthy cows didn't have. He would grow that up in a lab, introduce it into a new healthy organism. That new healthy organism would hopefully develop the same symptoms as the sick cows or sick animals or whatever it happened to have been. And then he would reculture out that organism, or try to, the same one as he found earlier, in his lab, and then show that that organism that he recultured out is really what caused these same symptoms, because he put it there into a previously non-sick cow, same symptoms developed, so I could also remove this organism out, which wasn't there to start with, so I put it there, it's caused the same symptoms, so this is what causes that. Really, really, really cool set of steps, Koch's postulates, and we still use them today. So, writing names in the microbiological world for microbiology. It's a big deal. I got a bunch of animals here for a reason. Those 2 birds are both robins. One's an American robin, one's a European robin. If I go to America and say I love robins, they're going to think of the European robin, not the American one. Totally different species of birds. They're both birds, but that's about the extent of how closely they are related to one another. They're like 15th cousins, twice removed kind of thing. A killer whale, it's a dolphin, not a true whale. An orca, that doesn't tell you what it's related to, so what's that mean? Mudbug, yabby, crayfish, lobster, crawdad, what are those things? So it depends on where you're from. Yabby is Australians' term for these things. You might not even know what people are talking about. And then you got buttercups, daffodils, so those different common names that people use for things and animals and plants and stuff aren't necessarily shared between different localities, different states, different countries. What if you don't even speak English? And that's going to get confusing. But animals and bacteria and things like that, living organisms also have something called a scientific name. It's called a binomial name. It's given two names that define who it's related to. Essentially, it's kind of like having a last name and a first name. The last name is first, tells you more who you're related to, and then the second name is specifically who you are. It's kind of like how Asian cultures write their names. The family names comes first, and this is essentially how we name these. So scientific names are very, very important because they're spoken across the world in Latin, and the same scientific name is in Russian, in Portuguese, and in English. They're all going to be the same. And they're not going to change where you're talking to people at. So if you say Orcanus orca in anywhere, they're going to know what you're talking about if they're a scientist that studies orcas. Whereas if you say an orca to somebody that doesn't speak English, they're just going to be lost. So scientific names are very important. Now in microbiology, they're even more important because in microbiology, it gets a little more aggravating because in this world, bacteria are often just named the same thing as their common name. So you see Bacillus cereus. Now, Bacillus cereus is a species of bacteria, but Bacillus is also a shape of bacteria. Staphylococcus is a shape of bacteria. Staphylococcus aureus is a species of bacteria. So that can get real confusing real fast. So being very important, being very aware, of how you write these or what you're reading can make all the difference when it comes to naming the microbiological world. So you can see here, they're written in a specific way. They're italicized or they're underlined, and that's to emphasize that it's a species. So if you just saw the word bacillus and it was not capitalized, it was not italicized or underlined, it would just be a shape of bacteria. But if you saw the word bacillus and it was underlined and capitalized, or capitalized and italicized, it would mean the species of bacteria known as bacillus and not the shape. So it's really important to make sure that you're paying super close attention to naming them and writing them correctly in microbiology more than any of the rest of the fields. It's important in all of them, but this one in particular. Now, microbiology also does abbreviate quite a few of these, E. coli, things like that. Now, E stands for Escherichia. There's quite a few other E's, Enterobacter, Enterococcus, so make sure that you know which E you're talking about because there's not just one of them. So just be aware of that. A couple of little terms to be aware of in the microbiology world. We have a microbe, which are things that we can't see. These are things that we may or may not talk about directly in this course, but you will hear tossed about from time to time, so we'll just talk about these a little bit. Infection, the presence of something living in or on you. I'm infected right now with E. coli in my intestine, as are you. They keep us alive, do very, very important things for us, but they're not killing us. So just because you're infected doesn't mean you're sick. Not always a bad thing. Signs are something that you're going to observe directly as a healthcare provider. So you as a nurse or a doctor or whatever would be looking at a patient and going, I see that you have a fever of 102. I can see you this bullseye-shaped rash. You have Lyme disease, whatever. Very, very, very useful because you can actually see what you're looking at and you can observe something. Whereas symptoms, on the other hand, are something that patients are going to tell you about how they feel. So my tummy hurts, or my head hurts. Okay, that's useful, but my head hurts. It's a big area here. Which part of your head? And they might not be able to communicate that clearly to you. So to kind of take that with a grain of salt, but it is useful. Inoculation, well, I'll use that term a lot in lab. We're going to inoculate new petri dishes. Humans receive inoculations and things like that. And that's introducing a bacteria or microbe into a new environment where it previously wasn't. So a disease. If you have a disease, you have an infection that's crossed over to causing tissue damage. That's not, you have a root cause that's not caused by you crushing your arm in the door or something like that. There's a living organism inside of you or some sort of acellular organism that's causing you to have some sort of tissue damage. Septicemia and bacteremia. Bacteremia, the presence of bacteria in the blood, not always a bad thing, usually not a good thing, but not always a bad thing. Whereas if they start to produce poisons toxins in your bloodstream, that's going to travel around through your bloodstream and cause tissue damage that way, and that usually is a bad thing. So both of those terms you hear a lot. And we'll finish up here a little bit with parasites and pathogens and things like that. So not every bacteria on the planet is out to do harm to you or every other living organism, and quite a few of them just live on or in us, and they don't do anything at all, other than just either live on us, and sometimes they're really important for keeping us safe. And those bacteria are called normal flora, chilling out, doing the thing in or on us. But occasionally you run into some bad guys, and those are pathogens. There's an opportunistic pathogen, which causes an opportunistic infection. And an opportunistic infection occurs when bacteria that live on your body can get into places where they're not supposed to be. It's kind of like if you keep the kid-proof locks on your door, your kid won't drink bleach from underneath the sink. But if you leave the lock off, they're going to go in there. That's just what kids do. And your body works the same way. The bacteria that live on your skin are fine as long as you don't have a cut. And most of the time, they're going to stay out. But occasionally, you can get a staph infection when you cut yourself because the bacteria can get in through your damaged skin kind of thing. So that's an opportunistic pathogen. And then you have true pathogens. And those are bacteria that are out to do you harm no matter what. If you come in contact with enough of them, they're going to make you sick. And that's just what they do. They can get inside of you. They can go around your body's defenses in some way, shape, or form. And they're going to make you sick. And then you got parasites, which we'll talk a little bit about later on. And those are organisms that live in or on you at your expense. They are taking either your food, your nutrients, or something away from you as a whole and harming you for their benefit. So basically, not very much fun on that one. That's really all there is to the introduction to microbiology, guys. If you have any questions on anything, please feel free to let me know. If not, have a good rest of your day.